I'm David Gerber. I'm a thoracic medical oncologist at UT Southwestern Medical Center in Dallas, Texas. I'm also study chair of the ECOG-ACRIN E4512 trial, which was a phase 3 trial comparing the ALK inhibitor crizotinib to observation in resected early-stage ALK-positive non-small cell lung cancer. To tell you a little bit more about that trial, I think it's worth taking a step back and discussing something called ALKemist. ALKemist is a large platform for early-stage clinical trials sponsored by the United States National Cancer Institute, or NCI. ALKemist stands for the Adjuvant Lung Cancer Enrichment, Marker, Identification, and Screening Trial. And ALKemist is enrolling thousands of patients with resected early-stage lung cancer, performing molecular testing, collecting clinical data, and then offering patients various adjuvant or post-operative therapeutic clinical trials according to tumor characteristics. That's really important for a trial like E4512, because E4512 studied ALK-positive early-stage lung cancer, which appears to be even less common in early-stage disease than it might be in metastatic lung cancer. For us to enroll 166 patients to the E4512 trial, that represented about 2% of the 7,345 patients that have been enrolled to date in the ALKemist study. E4512 is only one of a number of different therapeutic trials that have been done as part of ALKemist. There have also been studies done in EGFR mutant lung cancer and studies done looking at adjuvant immunotherapy and an ongoing study looking at adjuvant chemoimmunotherapy and determining whether combination concurrent or sequential is the best way to give those types of treatments. In terms of who we studied in the E4512 trial, as I mentioned, we enrolled 166 patients to this trial. Our target enrollment was 168, but we stopped enrollment at the point when the U.S. Food and Drug Administration approved adjuvant alectinib, a late-generation ALK inhibitor, for resected early-stage non-small cell lung cancer. That was in August of 2024 that happened. Of our patients, about 2/3 of them were women. About 60% of them were never smokers, which would be expected for something like ALK-positive lung cancer. And the vast majority were non-Hispanic white. Fewer than 10% of patients were Asian, and that makes our trial a very different population than you might see with other ALK inhibitor trials, whether early-stage disease or in the metastatic setting. The E4512 trial was very open-ended in terms of what the surgery requirements were, for example. So there was no specific surgical procedure required, no specific lymph node sampling, and adjuvant chemotherapy and postoperative radiation therapy both were allowed before starting treatment, but were not required. So what did we see? We saw that about 90% of patients had a lobectomy or pneumonectomy as their surgery. We saw that about 70% of them had extensive lymph node sampling. What did we see in terms of the efficacy? Our primary endpoint, and patients were randomized to 2 years of crizotinib, an early phase ALK inhibitor, or observation. The primary endpoint was disease-free survival in those cases that were centrally ALK testing positive. And in that population, we saw no difference in the disease-free survival between adjuvant crizotinib and observation in ALK-positive resected non-small cell lung cancer. The hazard ratio was 1.06. The p-value was 0.86. When we looked at disease-free survival in subgroups according to patient sex, cancer stage, or whether or not patients had received post-operative radiation therapy, we saw no difference there as well. A secondary endpoint was overall survival. There have been very few survival events or deaths in either arm to date, so it's very, very immature data, but we don't see any meaningful difference to date in terms of overall survival. The other secondary endpoint was safety. And in this trial, we collected data on grade 3 or greater treatment-related adverse events. And those occurred in 43% of patients receiving crizotinib. Only one of those events was actually grade 4. It was a stroke. The others were grade 3. And they were very similar to what we would expect from crizotinib from earlier trials. We saw gastrointestinal effects like diarrhea. We saw a very interesting visual toxicity where most patients taking crizotinib may see flashers or floaters or shadows when they start taking the drug, but with continued dosing, even if you don't stop it or adjust the dose, it goes away in most people. And other effects were edema or swelling. We had changes in liver chemistries and changes in blood counts. What struck me about this trial was that toxicity was the reason for people stopping treatment in over 1/4 of cases. And that's considerably more than we see in terms of what proportion of patients stop crizotinib due to toxicity in trials of crizotinib in metastatic lung cancer. Why might that be? Well, I have a couple of hypotheses. When people take adjuvant crizotinib, they may be taking it for longer than they take it in metastatic disease because the cancer hasn't recurred yet. Number one, so the longer you take a drug, the more likely you don't have symptoms from cumulative toxicity over time. And number two, we just have to wonder, is there a difference in not only a patient's motivation, but a medical oncologist's motivation, a treating physician motivation, to continue taking a drug, if it's, say, metastatic lung cancer, where you know if you stop it, the cancer is eventually going to start growing, versus a post-operative setting where you as an individual patient and you as an individual physician aren't quite sure what is the actual efficacy of what you're doing in real time.